For today, we are going to finish our discussion of the central nervous system. So last week, we talked about the spinal cord and neurons, and then today we're going to be talking about the brain. The brain is divided into two large components, two large sections. We have the cerebrum, which is this large section here. It becomes highly folded during development. It starts off as a neural tube, and then you'll get some swellings that will become the diencephalon and the cortex. So the cortex is this outer layer, really what people think of as being the brain. And during development, in order to increase surface area, it's going to grow and fold, and then it comes back, and then it folds back on itself, creating this temporal lobe, as well as the frontal lobe, parietal lobe, and occipital lobe. The lobes are named for the overlying skull bones. So that frontal lobe lies just deep to the frontal bone. We have our right and left parietal lobes that are just deep to the parietal bones. And then we have that occipital bone in the posterior. We have right and left temporal lobes. Okay. Then we have this smaller area down here. It looks kind of like a cauliflower. It also has lobes, but you don't need to know those three lobes because in our models, they don't really have real clear-cut delineations, but these are have lobes as well. But this entire structure is called the cerebellum. So we have our cerebrum and our cerebellum. When the brain does that folding during development to increase surface area, it creates structures. And you do need to know the names of these three and what they represent. So the gyrus is that surface portion of the fold. And so if you actually were to like put your finger kind of on the top of a brain model, what you're most likely going to be feeling is that gyrus. It's that top domed surface. We also have small grooves called sulcuses, and these we'll learn about the central sulcus and the lateral sulcus. These are going to usually divide lobes of the brain, and sometimes they'll also divide functional areas called domains. Fissures are very, very deep, and they will divide significant structures from each other. You do need to be able to identify the longitudinal fissure as well as the transverse fissure, the lateral sulcus and the central sulcus on both the brain model and the sheep brain dissection. Most of the structures will need to be able to be identified on both of those, with the exclusion of there's just a handful that I'll talk about that are on the brain dissection only. We'll start with the longitudinal fissure. Okay, so the longitudinal fissure is going to run lengthways, so it's very long, and it divides your brain into a right hemisphere and a left hemisphere. There is also a transverse fissure where the dura mater delves deep down in between and creates a significant separation between the cerebrum and the cerebellum. So the transverse fissure is across that posterior aspect. We also have two sulcuses. We have the central sulcus. So the central sulcus, I'll give you is actually pretty easy to see in the sheep brain dissection, and so I'll point that out again. It's if you, on the sheep brain, the longitudinal fissure kind of runs as the down bar of a T, and then the central sulcus will come out on the top of the T. The lateral sulcus is going to be here. It is very difficult to find in a sheep brain. We will only be looking at it with regards to the brain model, because we can actually mark it with a marker. But the lateral sulcus is going to divide that temporal lobe from the frontal and parietal lobes. Now, if we look at the brain model from an inferior view, so we flip the brain over and we look at it from the bottom, the first structures that you need to be able to locate are called olfactory bulbs. So as you can kind of guess from that olfactory, these bulbs are involved in the sense of smell. This one is not as obvious, but it's here. And then they are connected to the olfactory nerves. And on a brain model, this would have a Roman numeral 1, because olfactory nerve is cranial nerve number 1. So the olfactory bulbs are very distinctive on the sheep brain dissection. You can also find them here on the model. Then we will go to the chiasma. So the optic chiasma is the crossing over point of the optic nerves. So you have an eyeball here, and it is going to connect via this optic nerve here. And then you have another eyeball, and it's going to connect via this optic nerve. So the optic nerves often get cut off in the dissection. They look like little stumps in the brain model, but they do connect all the way out to the eyes. And then at this optic chiasma, some of the fibers will cross over, and then some will stay on the same side. This gives you that stereo vision. And then, so some of these nerve fibers will cross over and some will stay on the same side. Once we get down to beyond that optic chiasma, we get optic tracts. And so if you remember, a tract is a collection of axons in the central nervous system. So these are tracts, and they in a sheep brain dissection, they would be really, really white. They would glisten this beautiful white color because they're heavily myelinated axons, and they are going to continue to run deep through the brain into the occipital lobe for processing. So for lab, you just need to be able to identify the structures. You do not need to actually know their functions, but sometimes it's kind of helpful. Below the optic chiasma here are two structures that look like peas, and these are mammillary bodies. They contain cell bodies that are associated with the olfactory system. The next set of structures comprise the brainstem, and you do need to know what 3 structures are in the brainstem. You also need to know which structures are in the diencephalon. That's coming next. In the brainstem, we have the pons the midbrain, and the medulla oblongata. The pons is located as the middle of these three structures. And when you're looking at a model or at a dissection, it is the one that you identify first. The fibers deep in the pons run longitudinally, but on the surface, they run transverse. So you can usually identify the pons because it's this bulging structure, and then there are, looks like lines that are running across it. And those nerve tracts are communicating between different sections of the cerebellum. And then the deep fibers are communicating from lower to higher centers. But these are the fibers that you would see. If you go just above the pons, then you'll have the midbrain. So it's about an inch above. If you go below the pons, then you're going to have the medulla oblongata. And so the medulla oblongata is going to regulate your autonomic nervous system, and it is continuous with the spinal cord. So we just got through discussing that the brainstem is made up of the midbrain, the pons, and the medulla oblongata. In the next slide, we're going to be looking at these structures that are part of the midbrain. It is going to be a set of structures that comprise something called the corpora quadrigemina, but these are what are considered midbrain structures. The diencephalon is made up of the thalamus, the hypothalamus, and the epithalamus. And the most significant structure of the epithalamus is going to be the pineal gland, also known as the pineal body. So that's part of the diencephalon as well. The thalamus and the hypothalamus are, especially the thalamus, looks kind of like an Easter egg. And so it's round. And if you were to look inside of it, it would have these clusters of cell bodies called nuclei. So clusters of cell bodies in the peripheral nervous system are called ganglia. Clusters of cell bodies in the central nervous system are called nuclei. And both the thalamus and the hypothalamus are these structures that contain islands of cell bodies. So this is what we're going to be looking at next, is something called the corpora quadrigemina. If you are looking at a sheep brain, which you cannot do this with the human brain model, but if you are looking at a sheep brain from the back, from the top and then to the back, so this is the posterior aspect of that sheep brain, and then you pull down the cerebellum. So the cerebellum is roughly here. And you'll pull that structure down and away, and it exposes this set of structures here. The first structure that kind of sticks out at you, and it looks kind of like a leaf, is the pineal body. So the pineal body is the first structure that you would see. It is the one on top. It's by itself. Then there are two large round structures. Those are the superior colliculi. The superior colliculi are involved in visual tracking. So if you're watching, if you're catching a football and you're watching it go across the sky, if you're watching a bird, if you're watching cars as they are going by. So any of that visual tracking is the superior colliculi. Below the superior colliculi is a pair of inferior colliculi, but on the brain dissection, it looks like it's a single band. But in reality, it's two distinct structures. There's this divot in the middle, and it's going to be involved in auditory processing. If you take your 1, 2, 3, 4, so your 2 superior and your 2 inferior colliculi together, they make up the corpora quadrigemina, so a body of 4. And that's what those superior colliculi plus the inferior colliculi are called. Before moving on to brain dissection and the sagittal view, where we cut the brain in half and look at it, from that interior aspect, we're going to talk about the ventricles. So the ventricles are pools of cerebrospinal fluid. And what this model is showing is that if you plasticized that cerebrospinal fluid and then you pulled away the rest of the brain so that only those plasticized pools remained, that's what this model is trying to illustrate. There are 4 ventricles. The right and left lateral ventricle are considered 1 and 2, and they are directly underneath the corpus callosum. And they are going to be separated by a membrane called the septum pellucidum. So that's the membrane that separates the 2 lateral ventricles. The 3rd ventricle surrounds the thalamus. Okay, it's in a canal that is surrounding the thalamus. And then the 4th ventricle is at the opening of the cerebellum. It's this tail right here. Connecting the 3rd and the 4th ventricle is the cerebral aqueduct. So this cerebral aqueduct is the stalk, looks like the stalk on the model, and it is going to separate the 3rd, which looks like it has a hole in it, and then this little tail structure, which is the 4th. The 4th ventricle becomes continuous with that central canal of the spinal cord that we learned last week. So now I'm going to show you, there's 2 videos that are embedded in the PowerPoint. And the lab lecture video will not be ready until much later this weekend. And so if there is last week's lecture in that lab lecture video folder, this video, this lab lecture video won't be available in time for you to take your quiz. So if you want to review today's material, our last video that's embedded is by far the best video. It's like the best 8-10 minutes of time you could spend studying for the lab quiz or this topic. So the lab lecture video for today will not be available until probably Sunday. Just have a few things going on. So, and they're not posted separately either. So you'll need to download the PowerPoint in order to access the videos. Okay, so let me go to the video.
The sheep brain has the dura mater, the tough mother, covering it. So that's why you really can't see the brain yet. and we're going to have to cut that off.
Oh, sorry. It happened last time too.
The sheep brain has the dura mater, the tough mother, covering it. So that's why you really can't see the brain yet, and we're going to have to cut that off. Let's first take a look at the bottom of the brain, and you can see this is from the back of the eyeballs here, and we'll cut that off. And then right here is the pituitary gland. It's that little P-shaped structure right there. And you'll want to take a look at it because it might be the only time you ever get to see a pituitary gland. Also, you might notice on each side of the pituitary gland are some very fine blood vessels, and they're a radiator to cool the sheep's brain. That's important because sheep don't sweat, and they have to have a different way of cooling the brain off. And they do it with these blood vessels that radiate heat that was taken out of the brain by the blood. So let's flip it back over. The way I like to take the dura mater off is I like to make a little snip with the scissors, and I'm just going to snip right there. It's tough, you'll find that out. And now that I have the scissors in, I'm going to cut along the side. And I'm going to cut like this. I just have to peel this off, and you can see it starting to peel off here, and you're starting to see the brain underneath. I'm going to peel this back here. As I peel this part of the dura mater off, look for that dural sinus. That's where the cerebrospinal fluid flows back into after it's floated the brain. It's a vein that's right in the dura mater. I'm just going to keep peeling. Where the cerebrum is next to the cerebellum, the dura mater goes in between. And so I'm going to need to cut that. I'm going to take the scissors and go in like that, cut down. And, oops, I didn't cut quite enough. I think I got it. Almost. There. So I can pull that dura off and expose the cerebellum, the back half. So you see how we're getting the top of the brain uncovered. I'm going to flip it over, and now I need to gently peel this off the bottom side of the brain. But I want to be careful because I don't want to tear off the optic nerves, and the optic nerves came in right there. So I'm going to cut right there behind the optic nerves. We're going to have to take the pituitary gland off. So we have that off. And now I'm going to cut this part off. This is from the back of the eye, some of the fat and muscle behind the eye. So I have that cut off. I'm going to do the same over on this side. There, so that's off. And now I'm just going to trim a little bit more of the dura off like that. And now the dura is off the brain. When we're looking at the top of the brain, you can see the two halves of the cerebrum. And then if you use your thumb, you can gently separate the two halves and look down the longitudinal fissure, and you'll see the top of the corpus callosum. And we'll see that when we cut the brain into right and left halves. Then, if we turn it around, you can see the cerebellum here. If you gently pull the cerebellum down, you'll see the pineal sticking out like a tongue. You can see the pineal gland sticking out like a little pine cone or a little tongue. Let's take a look at the bottom side of the brain. Right here is the spinal cord, right here. This part of the brain is the medulla. So from here to here is the medulla. And then the pons, the bridge, is right here. And then this area right here is the midbrain. You might remember that this is the brainstem. And the brainstem's made up of midbrain, right here, pons, and medulla. This is the spinal cord down here. Right here we have the optic nerves. They still have a little bit of the dura mater on them because it's hard to get the dura mater off. You might be able to see the nerves of smell up front. It's a little hard to do that with this brain. I'm going to cut the brain in half now, and I think the easiest way to do that is to put it down on the pan. And I'm going to use a steak knife, and I'm going to follow the lines that this bottom half has. You can see what I mean? There's a kind of a line in the brain right there. You see some structures that kind of give you a line to follow with your knife. It's important to cut right down the middle between the right and left half. So I'm going to start like this. I'm going to try to just, I think I'm going to have to cut off this dura. I can't cut it with the knife. So I took off the optic nerves. That's okay. So now I'm going to cut right down the middle with the steak knife. Watch out for my fingers. And let's see how well I did. So now I have the two halves, and we can see some of the midline structures of the brain. Let me show you this one. This is the left half. So right here, you can see the pineal gland. And we saw it sticking up out at us when we looked at it from the back before we cut the brain. So there's the pineal. This right here is the corpus callosum. And if I poke into there, I'm opening up the space into the lateral ventricle. So the lateral ventricle is where I've got the tip of the probe. So there's the corpus callosum. You might see some of the blood vessels that make the cerebrospinal fluid. They're dark because they still have some blood in them. That's the choroid plexus, blood vessels that produce the cerebrospinal fluid. This oval structure right there is the thalamus, the switchboard for senses. And then below that, right here, is the hypothalamus. There is a little canal running right here, here it is. And that little canal is the cerebral aqueduct. And it carries cerebrospinal fluid down to the fourth ventricle, which is here underneath the cerebellum. One more thing I'd like to point out to you. I mentioned that the thalamus is kind of a ball right there. Underneath it, right here, is the hypothalamus. So those are the major structures.
Okay, so one thing I do want to point out on this view is you can see how white this optic chiasma is. So the optic chiasma is very, it didn't show very well, is very white. And then we have over here, so this is that optic chiasma. And then we have the mammillary body is over here. It's a little difficult to see on this view. But in between those two is going to be the hypothalamus. So the hypothalamus is this area that's located in between the optic chiasma and the mammillary body and is inferior to the thalamus. So the thalamus is very round, and then you have kind of this odd square rectangular substance or area below it, and this is the hypothalamus, and it's bordered by the optic chiasma on the towards the anterior side of the brain and the mammillary body on the towards the posterior side of the brain. But it does give you that good view. And then this is that pineal gland here. And then we have superior colliculi here and inferior colliculi here.
of the sheep brain. The sheep brain has the dura mater, the tough.
So just to review the structures that you should be able to identify on the sheep brain, we have the frontal lobe on the front, the occipital lobe on the back, and if you put your head right on top, these are going to be on top of your parietal lobes. So if you're looking at the longitudinal fissure, it's here, and then it tees into the central sulcus. So that central sulcus will divide the frontal lobe from the parietal lobes. And like the longitudinal, if you combine the central sulcus with that longitudinal fissure, you get this nice kind of cursive T structure. It shows up pretty nicely on the sheep brain dissection, and then very nicely on the human model because we have marked it in marker. Okay. This is the cerebellum back here. On a lateral view, you can see the temporal lobe. You can't really see the lateral sulcus very well in a sheep brain dissection. Usually we'll have, 24 students, we'll have a dozen brains or so, and we'll get one that looks great. And I'll be like, okay, everyone come over here and see this one. So it is here on the human model, it's easy to see because again, it's one that we've marked in marker. So cerebellum is the structure here, and the the transverse fissure is here dividing the cerebrum from the cerebellum. If you flip the sheep brain over, you have those olfactory bulbs kind of peeking out the front. They will then run down olfactory nerves. You have the optic chiasma in the middle with optic nerves running out to the eyeballs. Usually these are cut off, so if you see them, they're just little nubs on that kind of peek out here and here. And then from the optic chiasma, you have these glistening white optic tracts that are going to run down to the occipital lobe for processing. Mammillary bodies are the like little P structures here and here. The brainstem, remember, it is comprised of the midbrain, the pons, the medulla oblongata. You would find the pons first. It is going to be a bulging structure with lines that are showing you fiber tracks running this way. If you go 1 inch forward, you have the midbrain. If you continue further down before you hit the spinal cord, that would be the medulla oblongata. If you are looking at the brain from a dorsal view, so a back view, you pull down that cerebellum. This is only observable on the sheep brain model from this view. We can see these structures from a sagittal view as well, but they're best shown from this dorsal view. The pineal gland is the most superior structure, and it is singular, and we have 2 large superior colliculi and then a band of inferior colliculi below it. This is a sagittal view where we've cut the brain open. The notable structures, again, you should be able to name your lobes of your cerebral cortex. So the cortex is the folded area. I have the next slide will label the corpus callosum, but this is just to illustrate the 3 structures of the brainstem with, you locate the pons and So you locate the pons. And then if you go above that, that will be the midbrain. You go below it, it'll be the medulla oblongata. We also have the arbor vitae, which should kind of look familiar. Last week, we had our spinal cord. We had our deep gray horns surrounded by white matter. So that white matter then came up the spinal cord in columns. And now it's going to be running into, some of those fibers are going to run into the arbor vitae. And so in the cerebellum, you have this arrangement of white matter. These white tracks are deep, and then they are surrounded by two layers of gray cell bodies. So the gray matter is going to be superficial for the cerebellum and the arbor vitae. I'm just going to jump to, so this is the slide I was just showing you with, or trying to allude to, with those white tracks coming up and in, and then surrounded by superficial layers of gray. Other notable structures are the corpus callosum. The corpus callosum is this structure. This is called the fornix. So it's not one of the structures you need to be able to identify, but a lot of people go, oh, this is part of the corpus callosum, and it's not. So I just go ahead and label it for you so that you know what it is. The corpus callosum is what's called a commissural fiber tract. It communicates between hemispheres, so it allows the right and the left hemisphere to communicate. The fornix is going to allow communication with the limbic system. This is the lateral ventricle. Remember, you have two lateral ventricles, and they are separated by that membrane called the septum pellucidum. We have this round structure here, the thalamus. Then we have the, we'll have that optic chiasma that is going to be very glistening white. We have that mammillary body that is a deeper, fleshier color. And then in between those, we get the hypothalamus. If we were looking at a model, we would also, hanging from what's called the infundibulum, it's a stalk, we would have the pituitary gland hanging off. Okay, but it's been removed from this model. Again, with the sagittal section, we have our thalamus, we have our hypothalamus. This is the pons. So if this is the pons, then this is the midbrain here. And the superior colliculus and the inferior colliculus are midbrain structures. So superior colliculus is on top, inferior colliculus is on the bottom. And the pineal gland here is actually part of that diencephalon, so it's not a midbrain structure. And then the superior colliculus, the inferior colliculus, again, are going to be called the corpora quadrigemina. For these four structures, two superior and two inferior colliculi. Again, the lobes of the cerebrum. We have the pons is this bulging structure here. And so if you can locate the pons and you go anterior, this is the midbrain, inferior is the medulla oblongata ending in the spinal cord. This is the fourth ventricle here. And then it is going to be connected via the cerebral aqueduct to the third ventricle, which surrounds the thalamus. And then this structure is called the arbor vitae, the tree of life. It will have these white tracts deep, surrounded by 2 leafy layers of gray matter. This is just, again, showing you that this is the corpus callosum. We have the lateral ventricle is the space that's separated by the septum pellucidum, the thalamus, the hypothalamus, optic chiasma, mammillary body. This structure here is still the pituitary gland that's been left on this particular model. So some models and some dissections have the pituitary on and some have it off. So just thalamus to hypothalamus, optic chiasma, mammillary body, pineal gland, superior and inferior colliculi. We've got that third ventricle surrounding the thalamus, running through the cerebral aqueduct to that fourth ventricle. So this is my favorite video of the brain models. It is an excellent review because it gives you the same structures side by side with the human brain model and the sheep brain dissection. So if you have 10 minutes to review, download the PowerPoint and pull up this video.
You ever look at someone and wonder what is going on inside their head?
So that is it for that one. And then now we have just a few things to do, a few slides to go over. We've got 15 slides to go over for the lab test review. So half of these we went over last week when we were looking at the spinal cord and histology of the nervous system. The other thing I do want to bring up is that there are 2 pages in your lab manual. They are titled Human Brain Model and Sheep Brain Dissection. So they are here if you have an orange manual, or they're also located on that digital copy as well. And I would recommend just using some colored pencils to identify those structures. So don't forget to look at your lab manual. And then now we'll just go over the lab test review. And so these were from last week. And then there is a kind of a review of everything that we covered today. Okay. So this is looking at terminology. And so if we have so we'll just go ahead and I'll just have you chat these for the first few, and then we can go to our stamping feature. So what is the general name for this type of ridge? So it's the surface area part. Yep, very good. Okay, that is a gyrus. And then what is the general name for this type of small crevice? Yep, very good. And so this one would be the lateral sulcus. Okay. So now we can go ahead and, if you remember how to use that stamping feature underneath the annotate tool, go ahead and put a heart on the corpus callosum and a star on the cerebral cortex. It's the folded area. Yep, there you go. And then a check mark on the arbor vitae of the cerebellum, and a question mark on the pons. So the pons is kind of, yep, there you go. And then an arrow on the midbrain, and an X on the medulla oblongata. Yep. Usually when we do with the medulla oblongata, we put it a little bit more inferior, but it's fine. On the sheep brain, it's opposite the 4th ventricle. And then these are the 3 structures that comprise the brainstem, the midbrain, the pons, and the medulla oblongata. So if you will go ahead and put a heart on the hypothalamus. Started with a hard one. Yep, very good. Okay. An arrow on the thalamus. And one thing I have not mentioned yet is this little structure here is called a thalamic adhesion or a thalamic inclusion. It keeps the halves of the thalamus together. And on a human brain model, it's it looks kind of almost like a freckle, and it's always in the middle of the thalamus. On your thalamus, it might be offset, but in a human brain model, they always put the thalamic adhesion right in the middle. Okay, so let's go ahead and put a question mark on the septum pellucidum. And so the septum pellucidum is the membrane. This arrow here is pointing to the pituitary gland. It's kind of an odd presentation. We wouldn't present it this way. We'd present it more with the pink peanut model, like where it's hanging off, it looks like a pink peanut. And then go ahead and put a X on the spinal cord. Yeah, so this one's probably not my favorite, but okay. Then we are going to be looking at the ventricles right now. So we're looking at the cavities. And so let's go ahead and put a star at the fourth ventricle and a heart at the cerebral aqueduct and an arrow at the third ventricle. And I know it's not on here, but go ahead and put a question mark where the lateral ventricle would be. Yep, very nice. Remember, there's 2 of them, so that makes up all 4. So what is this pattern of white matter called? In the cerebellum. Yep, very nice. The arbor vitae, the tree of life. Then now we're going to be looking at lobes. So go ahead and put a heart on the frontal lobe, a question mark on the temporal lobe, a X on the occipital lobe, and it's not going to show up very well, but go ahead and put a star on the parietal lobe. Perfect. Okay, so we are going to be looking at a couple of sulcuses and a fissure. So go ahead and put a heart on the lateral sulcus. And an arrow at the central sulcus. It's not labeled, or it doesn't, yeah, there you go. And question marks at the transverse fissure. Very nice. And the fissure that we are not able to see that divides the right and left hemispheres is the longitudinal. Very nice, thank you. So go ahead and put a heart at the optic chiasma and a question mark on the olfactory bulb. This one's a little harder, but go ahead and star the pons. Yep, very nice. So that means that inferior to it, go ahead and put question marks at the medulla oblongata. Very good. What is this cord-like structure that goes from the optic chiasma to the occipital lobe? It is not the optic nerve, it's the optic tract. Very good. So the optic nerve is little, they look like little things that come off of it like this. And then the optic chiasma is in the middle, and then the optic tracts run to the side. So what is the pink structure that is missing? Or the spherical structure shown that's, yes, very good, pituitary gland. And then this is pointing to, if this is the optic chiasma, the coming off of it are the optic nerve. Very good. Okay. So now this is looking at a couple of midbrain structures and then a not midbrain structure. So the two black arrows are midbrain structures. Go ahead and put a heart on the superior colliculi. And then a arrow at the inferior colliculi. Yep. And then a question mark at the pineal gland. So the collective name for the superior colliculi plus the inferior colliculi is the very good, corpora quadrigemina. And the two structures that are with the black arrows are part of the midbrain. Okay, go ahead and put a star on the cerebellum. Oh yeah, it doesn't show up very well. A heart on the longitudinal fissure. An arrow at the transverse fissure. And a question mark on the occipital lobes. Yep. You guys got this. Okay, so go ahead and put a heart at the arbor vitae of the cerebellum and an X at the optic chiasma. A little harder. Yep, that's right. It's the glistening white one. An arrow at the thalamus. So the thalamus is going to be over 1. It's the round. You can't really see it. It's round. So this is, yep, that's going to be the thalamus. And then go ahead and put question marks at the corpus callosum. Yep. And then what is the membrane called? So the membrane is called the septum pellucidum, and that's going to separate those lateral ventricles. So this is looking at ventricles again. So go ahead and put a heart at the fourth ventricle, an arrow where the cerebral aqueduct would be. Yeah, very good. I didn't know you were going to get that one. And then go ahead and put X's kind of where you think the third ventricle should be. And it continues down. It's going to continue down and around the thalamus. And then a question mark at the lateral ventricle. Yep. Okay, so this is looking at the sagittal view again. Let's put a heart where the pineal gland is. Yeah, okay. And then a question mark at the pons. And an arrow at the, this is looking at the superior colliculus. So it kind of goes, yeah. And then just below that is the inferior colliculus. So the superior colliculus is this part here, and the inferior is this part here. This is not an ideal, like, because this got torn here, so it looks a little odd. Okay, so this one here is a very good view of structures you would find on the dorsal view. So if you put a heart at the pineal gland, and then arrow at the inferior colliculus. Yep, the bottom structure. And then question marks across the superior colliculus, which are those two, yep, in the middle. And then an X at the cerebral lobe of the cerebellum. Oh, I'm sorry. I meant to say the occipital lobe of the cerebrum. Yeah, thank you. Yes, and you're right. The cerebellum is down here. So I kind of, I could combine a few structures. Let's go ahead and put a heart at the olfactory bulb. And then an X at the, this is the pituitary gland. Nope, gotta go lower. The pituitary gland is going to be embedded in the dura mater. So that's going to give you a clue as to what you're looking for. If you see that this dura mater is still on the model, then that pituitary is embedded in it. In order to look at all those underlying structures, you would have to remove the dura mater and the pituitary gland. That's why you don't see it very often. Okay, so that is it for today.